All right, everyone, welcome to the last lecture of Bio 137. This will cover a couple of chapters. It's going to cover chapters 13 and 14, but like I've said a few times now, it's only going to be a very brief portion of each chapter. So the stuff that I do not talk about in this lecture, you don't need to know it from those chapters. So before I get to the not really attendance questions, I do want to talk a little bit about the autonomic nervous system, which is the second half of this lecture. Of all of the things that go from Bio 137 and carry over into Bio 139, it is the autonomic nervous system, the SNS and the PNS, that is going to be used the most. So make sure to hold on to these notes, review it if you need to, refresh right before you start Bio 137 and Bio 139, because they will literally discuss the autonomic nervous system in just about every single lecture next semester, and they will not review it. They will assume that if you got through Bio 137, you understand it. So make sure that you do and ask questions if you don't. Okay, so our not really attendance questions, the last batch of the semester. What is the sensory portion of a neuron? What is a ganglion? And what are the divisions of the autonomic nervous system? Go ahead and pause your video, try to answer those. Hopefully we've done that. So let's see what the answers to those are. First, what is the sensory portion of a neuron? This is the receptive part. Really what we're looking for here, this is the dendrites, but it could also be the soma. So the dendrites are really considered the sensory portion of a neuron, but the soma could be as well. What is a ganglion? A ganglion, remember, was a cluster of neuronal cell bodies in the peripheral nervous system. The same thing in the central nervous system would be called a nucleus, but in the peripheral nervous system, which is what we will be discussing today, it's called a ganglion, a cluster of neuronal cell bodies in the peripheral nervous system is a ganglion. And last, what are the divisions of the autonomic nervous system? Well, this is what we were just saying at the first slide. The autonomic nervous system is your sympathetic nervous system and your parasympathetic nervous system, typically abbreviated SNS and PNS. Remember from our intro to chapter 11, when we first started talking about the nervous system, we talked about the peripheral nervous system having two divisions. There was the motor division that was responsible for movement, and there was the sensory division, which was getting all of the information about your surroundings. You were sensing things. And in chapter 13, the sensory division is most of what we will be looking at in this chapter. So let's first look at those sensory nerves and see how we can classify them. And there's several different ways. The first way that we can classify these sensory nerves is by what type of stimulus are they sensitive to. One nerve typically will only respond to a certain stimulus type, and if there's a different stimulus, a different nerve will have to respond to that type. So first, let's look at mechanoreceptors. Mechanoreceptors are responsive to pressure or vibration or stretch. So it's something physical actually altering that nerve ending. Next, there's thermoreceptors, and I would say you can probably guess what thermoreceptors are sensitive to. Thermoreceptors respond to change in temperature, hot, cold, something like that. Photoreceptors These are found in the backs of our eye on something called the retina. Photoreceptors are responsive to light. They detect presence of light. And while we don't cover chapter 15, the special senses, I'll go ahead and say that there are different types of photoreceptors in our eye. We've probably heard of rods and cones. Well, rods simply detect presence of light. They detect black and white. They can tell if light is there and how intense that light is, whether it's bright, whether it's dim. Cones, on the other hand, there are different types of cones, and they detect different wavelengths of light. So that's our color vision. So photoreceptors are just responsive to light. Chemoreceptors respond to different chemicals. And when we talk about chemoreceptors, that could be something like our sense of smell or our sense of taste. Both of those respond to chemicals, either chemicals in our food or drink, or chemicals in the air. But within our body, we're constantly sampling our blood for how much calcium is in our blood, or what is the pH of our blood, or what is the carbon dioxide level in our blood. So there's different types of chemoreceptors to detect different types of chemicals. And nociceptors respond to pain. When something is hurting us, nociceptors are what's picking that up. This is a protective mechanism. We recoil from pain reflexively, so we have to be able to detect whether or not something could potentially harm us, and that's what nociceptors do. Well, we just saw how we could classify nerves based on the stimulus type that they respond to, but we could also classify them based on the location of that stimulus. Where is that stimulus coming from? Exteroceptors detect stimuli that are arising outside of our body. So when we see light, our eyes, those are exteroceptors because the light is arising from outside of our body. When we hear a car driving by, that sound is arising from outside our body, so that is an exteroceptor. Interoceptors detect things inside of our body. For example, when you've eaten a lot and your stomach feels really stretched out, that's interoceptors that are detecting that. Proprioceptors are kind of not really easily fit into either of the extero or interoceptor category. Proprioceptors detect the position of your body. So the way they work is they will detect the position of your joints, and they will detect things in your inner ear that tell if you are on balance or off balance. And all of these come together to let you know what position is your body in. If we were in a completely pitch black room and we could not see anything, if we couldn't even see our hand in front of our face, we'd still know which position our body is in, and that's because of our proprioceptors, the nerves that detect what position our body is in. Finally, we can classify based on structure of the nerve endings, the dendrites of the sensory neurons. For example, on this little table, we see non-encapsulated. So the dendrites are exposed. They're not wrapped by anything. This chart is on page 491 of your textbook if you want to see it in your book. So first of the non-encapsulated nerve endings, we have free nerve endings. Here the dendrites just spread through the tissue. There's nothing special about them. They're completely exposed. They spread through the tissue. Similar, we see a nerve ending that we have actually seen before back in chapter 5 with the skin, the integumentary system, We have modified free nerve endings, and we called these tactile cells or Merkel cells. So that's what we see right here. Remember, the Merkel cells themselves do not detect any sort of sensory information. They don't detect any stimulus. But just beneath them, we have a dendrite, and the Merkel cell passes information to that dendrite, which then sends a signal towards your central nervous system. So that's a modified free nerve ending, very similar to our free nerve endings, except it's got this Merkel cell there to pass information along to it. And we have hair follicle receptors, also found in our integumentary system. Here we see the dendrites wrap around a hair follicle. And when the hair shaft is moved, it kind of moves the hair follicle, which stimulates that neuron. If you've ever felt something kind of brush lightly the hair on your arm without actually touching your arm itself, if it just touches the hair of your arm, you can still feel something happening, like if there was a small bug crawling and it was caught in the hair of your arm. That will stimulate your hair to move, which in turn stimulates that hair follicle receptor, and we can feel whatever is moving that hair. But we can also have encapsulated nerve endings. Here, the dendrites are wrapped to some degree. They're not completely exposed like they were in the previous three examples. Our first two encapsulated nerve endings we've seen before. We saw back again when we were covering the skin, the integumentary system. We see tactile corpuscles, or we called them Meissner's corpuscles. Those were the ones that were really close to the surface of your skin, and they detected very light touch. A little bit deeper, we had the lamellar corpuscles, or the Pacinian corpuscles. Since they were deeper, they were not as easily stimulated, so they detected pressure, deep touch. We have something called bulbous corpuscles, or Ruffini endings. These are found a little bit in your skin, but more often we tend to associate these with your joints. These are found in your joint capsules, and they detect movement of your joints. When your joints move, that capsule is scrunched and stretched, and these detect that movement of your joint and convey to your brain exactly what your joints are doing. So you get a sense of what position or what movements your body is experiencing. In the muscles, we have something called muscle spindles. And here, the nerve endings wrap around muscle fibers. And as your muscles are stretched, those muscle fibers get thinner. When your muscles contract, those fibers get thicker. And we can detect how thin or how thick those muscle fibers are based on these muscle spindles. And that conveys also to your brain what position your muscles are in. Are they contracted? Are they relaxed? Our tendon organs, also called Golgi organs, not the same as a Golgi apparatus from chapter 3, but Golgi organs or tendon organs are really similar to muscle spindles, except they wrap around tendons. So they detect how stretched or how unstretched a tendon is based on how skinny or fat that tendon is. And that also conveys to our brain what position our muscles are in based on what position those tendons are in and how stretched they are. And finally, we have joint kinesthetic receptors. Joint kinesthetic receptors, also very similar to the Ruffini endings. They are found around our joints, and they detect movements of joints and give us an idea of, are we moving, how are we moving, which part of our body is moving, and also conveys an idea of what position our body is in. Now let's look at a slightly larger scale. Up to this point, we've been talking about nerve endings. So really, we were talking about the level of the neuron. We were looking at dendrites. But now we're going to talk about the structure of a nerve. And remember from chapter 11, a nerve is not a neuron. A nerve is actually bundles of neuron parts. Specifically, we're looking at bundles of axons in the peripheral nervous system. That was our definition of a nerve from back in chapter 12. And this is going to be really similar to what we learned about the structure of a muscle in chapter 9. The words are going to sound really similar to the terms that we used with muscles. So first, what we see in this picture here is a nerve that we have cut so that we can see inside of it. And the nerve is wrapped in this connective tissue sheath called the epineurium. Epi on the surface of, neuri pertaining to a nerve. So the epineurium wraps a nerve. And inside the nerve, we can see bundles, and those bundles are called fascicles, exactly like they were in a muscle. So fascicles are found within the nerve itself. And here we pulled a fascicle out, and we can see that it's got a connective tissue wrapping also. That's called the perineurium. So a fascicle is wrapped in a perineurium. And inside that fascicle, we see smaller structures still. These are our axons. And axons have their own wrapping, the endoneurium. So axons are wrapped in an endoneurium. Several of them together make a fascicle, which is wrapped in a perineurium. And then several fascicles together make a nerve, which is wrapped in the epineurium. Now, in between the fascicles, that's where we find our blood vessels. Nerves are relatively large, so they need their blood supply, and that's found in between the fascicles. But let's go back to the level of the axon. So here's our axon wrapped in an endoneurium. And remember, some axons were myelinated. They had that myelin sheath wrapped around them. If it is a myelinated axon, the myelin sheath is found inside of the endoneurium. So we peel the endoneurium back, and we can see the myelin sheath, and then the axon all the way in the middle. What happens if a neuron gets damaged? How does it regenerate? Well, we almost answered that question in chapters 4 and 11. We have previously said that if a neuron dies, it's gone for good. They are amitotic. They do not go through mitosis. But this is not saying what happens if a neuron dies. This is saying what happens if a neuron gets damaged. So what is it that determines if it's going to die or if it's going to be able to regenerate? It depends on which part of the neuron has been damaged. Remember, in the cell body, the soma, that's where the nucleus is, and that's where the majority of the organelles are found. And if damage occurs at the soma, it's a pretty sure bet that that neuron is going to die and it will be gone for good. But in this picture, we're seeing what happens if there's damage to the axon. Well, if we damage the axon, but the soma remains intact, we can actually regenerate or regrow an axon. If we've ever heard of someone who broke their neck or broke their back and they were paralyzed, and it's a matter of, are they going to be able to walk again? Okay, sometimes they're not able to. Sometimes they're paralyzed and they're paralyzed for life. But sometimes you will hear someone regained movement or regained feeling in their legs. So what happened? In that case, if someone is able to start moving again or start feeling again, it sounds like they probably only damaged axons, and it was just a matter of time before those axons regrew. So let's look at how an axon can regenerate. What we see here in this picture is somewhere along the axon there's damage, and now we see that axon has been severed. And when that happens, the end of the axon closest to the soma, that seals off. But the end of the axon further from the soma, that begins to degenerate. It's not attached to anything where there is a nucleus, so it will die off. And from that point, this end of the axon that's closest to the soma can regrow, and that's what we're going to see here. After the damage occurs, the Schwann cells kind of clean up the remaining bits of that myelin sheath, and macrophages begin to arrive to clear up cellular debris, things like the bits of axon that were there. So macrophages are cleaning up the debris as Schwann cells are cleaning up the myelin sheath. Once this cleanup is pretty much over, the Schwann cells begin to reorganize themselves, and they start to form this hollow tube called a regeneration tube. And that regeneration tube ultimately is where the axon is going to be found. So once that regeneration tube forms, that axon begins to sprout filaments that grow through that regeneration tube. And when they reach their full length and get to where the axon terminals originally were, those filaments begin to fuse together and now are back to resembling an axon. And once that axon has taken shape, the Schwann cells begin to wrap themselves around that axon in that jelly roll-like fashion we had seen back in chapter 11, and now we have our myelin sheath back in place, and we have an axon back to its original form. In lab, when we looked at our spinal cord model, we saw that the nerves arose between two vertebrae, and we named those spinal nerves after the vertebrae where they arise. So just like we called the vertebrae things like C1 or cervical 1, C2, C3, or T5, T6, or L4, something like that, we give the same names to spinal nerves. And as these nerves leave the spine and they go out towards whichever location they're serving, they branch and they cover more and more area of the body. But they follow a pattern, and that's what we're seeing here with dermatomes. Anywhere where we see one numbered spinal nerve, for example, here in the neck, we see C4 at the lower neck, very upper part of the shoulder, and it wraps around to the back of the neck. That whole area of the skin is supplied by C4 nerve. C5, we can see also, it expands down the arm. Or C8 expands down the other side of the arm. Down towards the legs, L4 wraps around the front of the leg all the way down to the inside of the foot. So what this is telling us is, in a clinical setting, if someone has damage, say a pinched nerve or something, we can narrow it down. if we take something and we poke this portion of the arm right here, and the patient doesn't feel any sensation, that's a good guess that C5 is damaged. Similarly, if we're going to have to do some sort of surgery and numb a nerve, if we know where the surgery is going to take place, it helps to narrow down which nerves we need to actually block sense from. Now let's move to chapter 14, the autonomic nervous system. And as I said in the beginning, this is the chapter that is most used in Bio 139. We're going to refer to ANS constantly in Bio 139. We're going to use PNS, SNS, and it is just expected that you know what that means and what their purpose and function are. So first, let's look at the autonomic nervous system as a whole. Remember, the ANS was the part of your nervous system that was responsible for all of the things that kind of keep your body going in the background without you having to think about it. Things like your breathing rate, your heart rate, your blood pressure, your digestion, things like that. And it was divided into two parts, and one of these parts was always turned on while the other was turned off, and sometimes they would flip. So those two halves were the parasympathetic nervous system, the PNS. We summed that up back in chapter 11 as rest and digest, relax and reproduce. This is what's in control when you are eating a snack or you're at home and calm and watching TV. So when you are relaxed, when you're sitting at home watching TV, you have a very low blood pressure. You have a slow heart rate. You're not breathing very fast at all. If you're eating a bag of chips, then you're also digesting those chips in your digestive system. But what if while you're sitting there watching TV, someone kicks your front door in and starts yelling at you? Well, you're not going to be so calm anymore. This is actually when your PNS turns off, but your SNS, your sympathetic nervous system, takes over. Your sympathetic nervous system, SNS, is your fight or flight response. This is when you fight for your life or run for your life. And if you can imagine yourself in that situation, some of the things that happen are your heart rate spikes. It starts beating really fast. Your blood pressure goes up. You start breathing quicker. Your pupils dilate to let more light in so that you can see better. But at the same time, you're not so much concerned with digesting that food that you just ate. So when your SNS turns on, things like your digestion turns off or turns down. So your SNS or PNS are on alternate sides. When one goes up, the other goes down. And they're laid out a little bit differently structurally. If we were to look at these, there's always going to be two nerves or two neurons, one right after the other. And one of them arises in your central nervous system and runs to a point where it meets with the second neuron. And since we're talking about the peripheral nervous system, remember the ANS is a part of your peripheral nervous system. Well, where that first neuron ends, there's going to be a group of cell bodies where the second neuron begins. Think back, what was a group of cell bodies in the peripheral nervous system called? It's called a ganglion. And the ganglion is where the second neuron begins. So we have a first neuron that goes from the central nervous system to the ganglion. Let's call that the preganglionic neuron. And then we have a second neuron that runs from that ganglion to the organ that it is going to innervate. Let's call this the postganglionic neuron. So in the PNS and the SNS, we have a preganglionic neuron that arises in the central nervous system and a postganglionic neuron that arises at the ganglion. The layout of the PNS and the SNS are different. First, let's look at the PNS. The PNS arise from either the brainstem or the sacrum. So that preganglionic neuron comes off of either way up here at the top of the spine or way down here at the very end. The preganglionic neuron in the PNS is very long. It runs from the central nervous system all the way to the organ that it is going to innervate. The ganglion is actually located at the organ itself. So the postganglionic neuron in the PNS is really, really short. It only has to run from the ganglion to the organ, and since the ganglion is already at the organ, it doesn't need to be very long at all. In the SNS, it's a little bit different. The preganglionic neuron arises either in the thoracic spine or the lumbar spine. And right next to the spinal cord, there is this row of ganglion, and the ganglia there are right next to the spinal cord. So let's look at our next slide and see how that actually looks. Here's a portion of our spinal cord, and it shows one of the vertebrae. So here's our vertebrae right here, spinal cord inside of it. And we can see running along the side of that vertebral column this other chain right here. And this lump, that's called the sympathetic chain ganglion. So this is where the ganglia for the sympathetic nervous system are found. And that means that the preganglionic neuron in the sympathetic nervous system arises in the central nervous system, exits the spine, and right here, just next to the spine, is where the ganglion is. So the preganglionic neuron in the sympathetic nervous system is really short. It only needs to run from the spine to just outside of the spine. But the postganglionic neuron begins at that ganglion and then has to run all the way to whichever organ it's innervating. So the postganglionic neuron is really long. So to sum that up, preganglionic neuron in the parasympathetic nervous system runs from either the brainstem or the sacrum, the central nervous system, all the way to the organ that it's going to innervate. That's where the ganglion is found in the PNS. And then the postganglionic neuron is really short. It runs from that ganglion to the organ. But in the sympathetic nervous system, that arises in either the thoracic or lumbar spine. The preganglionic neuron of the SNS is really short, running from the central nervous system to the sympathetic chain ganglia. The postganglionic neuron in the SNS runs from that ganglion all the way to the organ that is going to innervate. So how do your sympathetic nervous system and your parasympathetic nervous system have such opposite effects on the different parts of your body? That comes down to which neurotransmitters, or in one case we're going to see in a moment, hormones, are released. Which ones are released from where, and what are the effects of those hormones? Well, before we talk about the autonomic nervous system and its neurotransmitters, let's look back at the somatic nervous system. So this is really what we looked at in chapter 11 and chapter 9. Here we saw that there was a neuron that was very, very long, and it went to a muscle, and at the muscle, it released the neurotransmitter acetylcholine, ACH. And that ACH caused your skeletal muscle to contract. Well, it caused an action potential, and then that action potential led to a contraction, but you see what I mean there. Well, let's look at the parasympathetic nervous system. What we see down here, here's our very long preganglionic neuron. Here's our ganglion, and our very short postganglionic neuron. In the parasympathetic nervous system, the preganglionic neuron releases acetylcholine, and the postganglionic neuron releases acetylcholine, and acetylcholine is what does things like slows down your heart or causes your digestive system to move food through or causes you to breathe slower. That's your parasympathetic nervous system. Well, let's look up here at our sympathetic nervous system. Here's our very short preganglionic neuron. Here's our ganglion, and here's our very long postganglionic neuron. Our preganglionic neuron releases acetylcholine. So every time so far in the somatic, in the preganglionic parasympathetic, and in our postganglionic parasympathetic, and now in our preganglionic sympathetic, this is acetylcholine. That's all that we've talked about so far. Here's where the difference comes in. In the sympathetic nervous system, the postganglionic neuron releases a different neurotransmitter. It releases norepinephrine, sometimes abbreviated NE, sometimes called norepi. So norepinephrine, norepi, NE. This is what causes that very quick increase in heart rate, that very quick spike in blood pressure, that very quick burst of, oh, I got to breathe really fast. That's what norepinephrine does, and it's extremely fast acting. But if you've ever noticed when you get a scare, if your friend ever thought it'd be hilarious to jump out from around the door and scare you, you know you do get that very quick burst of fear. That's the norepi. But it doesn't very quickly go away. You can tell that there's a little bit of trembling that goes on for a while. It's long lasting. And as we talked about in chapter 11, neurotransmitters, they're fast acting, but they are not long lasting. So what's the mechanism to keep that prolonged fear going? Well, the sympathetic nervous system actually has another method also. Instead of having this preganglionic and postganglionic, we have one neuron that goes to a gland. This is your adrenal gland. And this neuron in your sympathetic nervous system releases acetylcholine onto your adrenal gland, and the middle part of your adrenal gland, the adrenal medulla, releases some norepinephrine, but also epinephrine. So we say it releases epi and norepi. And epi is the same epi that we hear about in EpiPens. Epinephrine and norepinephrine are released into your bloodstream. This is a little slower acting, but it keeps that effect going longer. So when your adrenal medulla releases epi and norepi into your bloodstream, we get that prolonged sense of fear, that prolonged, ready to fight or ready to run. So the sympathetic nervous system has this very quick layout with the norepinephrine, and a very long-lasting but slower-acting layout with the epi and norepi into the bloodstream. Now, we actually have heard of epi and norepi acting in this way before. If you've ever heard of, there's always the story of mom has a baby and the car somehow or another is on the baby. I don't know how that ever happens, but we've all heard that story. And what does mom do? Well, she hulks out. She lifts the car up because she has a burst of adrenaline. Well, adrenaline, that's epinephrine. Epinephrine is adrenaline. It comes from the adrenal gland, adrenaline or adrenaline. Adrenaline, epinephrine, does not give you super strength. It does not make that mom able to lift the car because it gives her extra strength. No, we could all go out and lift a car, but we don't do that. And the reason is, if we try to lift a car, it hurts. Maybe we don't realize that it hurts. We lift the car and then we say, no, it's too heavy, I can't do it. But that's your nervous system saying, if I keep doing this, I'm going to damage myself and it's going to hurt. So you stop before you lift the car. But your sympathetic nervous system dulls your sense of pain. So instead of trying to lift the car and stopping when you realize I can't do it, mom lifts the car and doesn't even realize that it hurts. Later on, when all of this epi and norepi wear off and her parasympathetic kicks back in, we don't ever hear this part, but mom will be in a lot of pain. She's probably damaged some muscles, but she saved the baby. And that's part of what the sympathetic nervous system does. That's your fight or flight response. Mom fought.
All right, so that brings us to the end of Unit 4. Up next, we have our 4th lecture exam. And right after that is finals week. So you need to be ready to take exam 4 over Unit 4, and then our final exam, which will cover everything from lecture from the first day of class to the last day of class. So it will cover everything that we have talked about in this class so far. And that's why I have a little bit of older material on each exam, is because I want you guys to stay in the habit of reviewing old material. Hopefully most of it is still pretty fresh in your head now because of that, and you're not all of a sudden being tested over material that you haven't thought about for several weeks, several months. That brings us to the end of this semester. We are done. There is no more new material. That is the end of the class, but a couple of things for Bio 139. So in Bio 139, it is not a deeper look at what we've already done. Bio 139 is really just the second half of a really long class. It's going to cover pretty much the second half of your textbook. So make sure to continue to ask questions. As we go towards a break, I do not take time off. If you are the type of student that is reviewing over break, reach out and ask those questions. Even if you have another instructor next semester, I am always willing to help. Keep my number and my email handy. When you have questions, reach out and ask me anytime. I still love to talk about science, even with people that are not my students. So hopefully you're ready to move on. Be sure to review anything, especially those things that I say come back in Bio 139. As we go through our lectures, I was saying this is going to come back in Bio 139. Hopefully you made some notes about that and kept that handy. If you have me for Bio 139, I do send out an early announcement letting students know good topics to refresh on shortly before the start of class. So keep an eye out for that if you do have me for Bio 139. And again, if you don't have me, I'm still always willing to help. All right, that's the end of our content. That's the end of all of our lectures. All we have left are some exams. Good luck on those. Take care, and hopefully I'll see you all in Bio 139.